welcome to our presentation on our paper. I'm Danny, and I'm with Sam and Ben, and we're going to be presenting on the paper Uncoupling Protein 1 and sarcolipin are required to maintain optimal thermogenesis, and loss of both systems compromises survival of mice under cold stress. So just to introduce a couple things, as we know, endotherms are animals who rely on the internal temperature of the body and basically just homeostatic mechanisms to maintain temperature. And ectotherms are things like reptiles and amphibians that rely on external environments to regulate the temperature of their body. So endotherms are what we are. So how do we and mice maintain body temperature below thermoneutrality? As you can see on this little figure right here, when mice, their normal thermoneutral temperature is going to be 30 degrees Celsius, and the standard temperature would be 21 degrees Celsius, so that's what would feel normal to us, but that actually gives them a state of cold stress, which we'll talk about a little bit later on. But whenever we experience body temperature below thermoneutrality, that basically means that we're cold. And one of the things that happens whenever we're cold is we induce something called facultative thermogenic mechanisms, and these are something that is switched on and off by the nervous system. There's something also called an obligatory thermogenesis, but we're not really going to talk about that much in our paper, but that's basically like your fundamental baseline metabolic processes that produce heat and whatnot. So for facultative thermogenic mechanisms, your first line of defense is going to be shivering thermogenesis, and that's exactly what it sounds like. I'm going to talk about that on the next slide, but what you need to know for this paper is that shivering thermogenesis for too long or prolonged shivering is detrimental to the body. So your body will induce something called non-shivering thermogenesis, and that's what we refer to NST in this paper. So in shivering thermogenesis, as we know in most of our classes, any bio class or anything that you've talked about, homeostasis and how we are constantly trying to maintain homeostatic environments. But one of the things that happens when we're cold is we begin to shiver, and how that happens is our body senses cold through receptors called C fibers and alpha delta fibers, and these really aren't super important to the paper, but they're basically unmyelinated and myelinated fibers with low or fast conduction velocity. So once your receptors feel like that coldness, they trigger a bunch of signals, and that basically tells your nervous system to release signals for your skeletal muscles to contract and relax super rapidly, and that's where we get that shivering phenomenon. So non-shivering thermogenesis is what happens whenever you can't sustain shivering thermogenesis anymore, and this occurs a lot in brown adipose tissue, mainly in adipose tissue, but that's something we'll talk about further in the paper. But this non-shivering thermogenesis is mediated by uncoupling protein 1, which is super important for this paper, and that's what we will refer to as UCP1. And so what this means is that whenever we, or in this case mice, are in need of extra heat, uncoupling protein 1 facilitates thermogenesis, non-shivering thermogenesis, by dissipating the mitochondrial gradient. So basically this is kind of like UCP1 is kind of like that FCCP that we see in prior papers. We'll get into that in a little bit further, but uses the chemical energy of ATP to make kinetic energy, and that means that heat leaves as a byproduct. Basically, here's just a little figure of what thermogenesis looks like in our bodies. It's basically this big feedback loop. So you feel this cold temperature, whether that's skin temperature, brain temperature, visceral temperature, and that goes to your hypothalamus. And so that will release certain hormones, whether that's somatic, which triggers that shivering thermogenesis, or your sympathetic nervous system, which will trigger that brown adipose tissue or beige thermogenesis, vasoconstriction. So your core body temperature will rise, and then it'll feed straight back. And then what is brown adipose tissue? This can be really important for the paper, but basically, as we've learned in class, adipocytes are cells specialized for fat storage. So your brown adipocytes is something that we see a lot in mice. We don't really see it in humans. We see them in babies, and for a really long time they thought it was just babies, but researchers found that we do, as like adults, have a little bit in our shoulders and our back, as you can see right here. These little sites from this PET CT scan. So brown adipocytes are rich in mitochondria and rich in fat droplets, as you can see. So they have lots of mitochondria and lots of fat droplets versus those white adipocytes. So these have one fat droplet and very, very few mitochondria, hence why they're white. Because, as we know, mitochondria are rich in iron, and iron is brown. And yeah, so the white adipocytes don't have that mitochondria that would reflect that brown color. So brown adipose tissue is a regulator of non-shivering thermogenesis, and that means it can be activated to oxidize fatty acids to maintain body temperature. This will be talked about a little bit more in the future of the paper, but is uncoupling protein 1, UCP1. As we can see right here, UCP1 is an inner mitochondrial membrane protein, and it functions a lot like FCCP does, like we've seen in prior papers, and is highly expressed in brown adipose tissue. So when your body is in a state of non-shivering thermogenesis, you know that we are cold and we're trying to maintain that homeostatic thermoneutrality, trying to gain more heat just so we can have a neutral core body temperature. So what it does is, again, functions like FCCP, and it generates heat by dissipating the mitochondrial proton gradient. So what does that mean? Instead of those protons going to ATP synthase and starting making ATP, it goes through UCP1, uncoupling protein 1. And you can kind of think of it like a proton link. So the energy dissipated in the gradient is kind of trapped in the synthesis of ATP. So UCP1 uncouples the O2 consumption, which we know that O2 is a universal acceptor from oxphos. So what it does by uncoupling the O2 consumption from ATP synthesis, it kind of short circuits the mitochondrial process. And once those protons come in and are kind of leaked out, heat is generated as a byproduct. So what is sarcolipin, SLN for short? This is a gene, and it is a regulator of sarcoplasmic reticulum calcium ATPase, and this is SERCA for short. But basically, regularly, calcium goes from the cytosol to the lumen of the sarcoplasmic reticulum. But sarcolipin uncouples calcium transport from ATP hydrolysis, and by doing that, you uncouple the respiratory train from the ATP generation, which sounds kind of confusing, but in the grand scheme of things, it basically just promotes this futile cycle of this pump that is in the membrane. And whenever you increase this futile cycle, this means kind of like a pointless cycle. It's not really doing anything. This increases ATP hydrolysis, and heat is also a byproduct, like we are seeing in UCP1. So, just a little bit more introduction. I know this has been a lot, but this is a lot of content that we haven't covered, so we just want to make sure that you're covering the basis of everything. But what is red muscle versus white muscle? So as we can see right here, red muscle has high mitochondrial content, thus all the red, because we know that if you have a high mitochondrial content, then you're rich in iron and myoglobin. And so red muscle is not very present in the sarcoplasmic reticulum, but that means we have a slow rate of contraction. So these are things such as your extensor muscles. We're not really going to talk about mixed muscles too much, but white muscles contain low mitochondrial content. So this means we have less mitochondria, less myoglobin, thus no redness. And white muscles are more abundant in the sarcoplasmic reticulum, and they have a fast rate of contraction, and they can perform anaerobic oxidation, and so accumulation of lactic acid is much quicker here than the red muscle, so we don't get a lot of lactic acid buildup right here. And this is things such as your eye muscles. So knowing examples of that, it probably wouldn't be a good thing if we had a buildup of lactic acid in our eyes. So, more introduction, browning of white fat. This is going to be important later on in the paper, but As we know from non-shivering thermogenesis, when we have brown fat, brown adipocytes, that means we can upregulate mechanisms to produce more brown fat. And this is important because we know that non-shivering thermogenesis uses brown fat through the UCP1 to generate heat. So there's this process called browning of white fat, and it's a little bit misleading, truly, because we're not really making brown fat from white fat. We're just kind of making a phenotypically similar molecule that has similar characteristics that can be used for the same function. So when you have white fat, you have this large lipid droplet. We don't have a lot of mitochondria, so they don't really serve a purpose in non-shivering thermogenesis. So through a process, the most studied one is going to be through norepinephrine, but this is also a relevant topic right now, so it's constantly building. So there's a bunch of different research going into the browning of white fat, but one of the most studied methods we're going to talk about later too is norepinephrine. So whenever norepinephrine is released from the sympathetic nerve, it acts on the beta adrenergic receptors on the surface of adipocytes, and that basically encourages this process to happen, where you form like this bright adipocyte. So bright is brown and white, kind of like a beige adipocyte, and it's phenotypically similar to the brown adipocyte. So if we can see here, we see a lot of mitochondria, we see lots of lipid droplets, and then we see heat being produced. We also can see right here these little red circles right here are going to be those uncoupling proteins that are going to be important for generating heat. So phenotypically, these are very similar molecules, but they are not the same. So now that we know all this information that we've covered so far in this very long introduction, what is the purpose of doing this experiment? So as we covered before, brown adipose tissue is really important for non-shivering thermogenesis, and that's mediated by that uncoupling protein 1 that we covered earlier, and that occurs by dissipating the mitochondrial gradient, and that produces a proton link where heat is a byproduct, therefore heating up our inner temperature to our thermoneutrality. And anytime we reference UCP1, we're referring to that uncoupling protein. So when UCP1 is knocked out, anytime we say UCP-KO, that's what we're referring to, knocked out. This means that that protein is not there, that gene is not there. Anytime a KO is after a certain gene or protein, that means that it has been knocked out in some way. For this UCP-KO, it has been knocked out through a double crossover through heterozygotes, but that's not super important for understanding the paper. But the purpose of this entire experiment is that when this uncoupling protein 1 is knocked out in mice, some lose the ability to perform non-shivering thermogenesis and some don't. Now, this is confusing because we know that UCP1 mediates that brown adipose tissue, and brown adipose tissue is that regulator for non-shivering thermogenesis. So, if we don't have UCP1, if it's knocked out, why can some still do non-shivering thermogenesis? So the purpose of this entire paper is that UCP1 independent, so whenever UCP1 isn't there, UCP1 independent non-shivering thermogenesis is not well understood. So the researchers are thinking something else is playing a role, but what is this? They're thinking that in this paper, they'll elaborate this on a little bit more, but could this be sarcolipin, this SLN, that's what we're referring to? Could SLN be compensating for UCP1 independent non-shivering thermogenesis? So, looking at figures 1A through 1C, this is titled Muscle of Cold Adapted UCP1 KO Mice, and anytime we talk about UCP KO, that means uncoupled protein 1 knocked out. If anything's followed by KO, that means knocked out. The protein's not there, the gene's not there. So this was done in the quadriceps and the soleus, and just to tell you what those are real quick on this slide, the soleus is that lower leg muscle in mice, and quadriceps is basically the same things that we see in humans. It's that upper leg muscle. what they did is they started at 28 degrees Celsius, and this is already a state of cold stress for the mice. Like I said in the first slide, their state of thermoneutrality is going to be about 31 degrees. So they started at 28 degrees Celsius and then reduced to 4 degrees, and then maintained that for about 10 days. And this allowed them to see how the mice reacted to cold adaptation. And so what they did is they did cell staining and then a Western blot. And so what they saw whenever they did the cell staining in the wild type and the UCP1 KO wild type is going to be just like a control, the one that has the protein in it. They see a increase in muscle redness and an increase in myoglobin content, like we see right here. There's barely any in that wild type, and then there's an increase in that UCP1 KO. And there's an increase in redness, an increase in redness in comparison to the wild type, even though I know that's kind of hard to see. But what they saw is that in the wild type, whenever they did this cold adaptation, is that there was no sarcolipin, the SLN, being expressed. But whenever they knocked out that UCP1 KO protein, or whenever they knocked out UCP1, there was an upregulation of sarcolipin. And just to remind you, GAPDH is going to be that control for both of these. Just make sure that we have consistent results, even though this isn't super consistent, but it's okay. Anyways, they also see this in both muscles, the quadriceps and the soleus. They see an upregulation in sarcolipin in comparison to the wild type, where there's barely any, even though we do see a little bit right there. And then in the diaphragm, we were trying to figure out why there was no upregulation of SLN. We've come to the conclusion that they said in the paper, and we're just going to go with it, that the diaphragm already expresses high levels of sarcolipin, so it's really unchanged here. But what does this entire thing mean? We see an increase in muscle redness, and we see an increase in myoglobin content, and we see an increase in the sarcolipin expression whenever that UCP1 is knocked out. This means that the UCP1 knocked out mice use sarcolipin to adapt to the cold exposure, and this increase in sarcolipin expression could potentially compensate for the loss of UCP1 function or the loss of brown adipose tissue. This again is just showing you the quadriceps and the soleus.
So moving into figure 2, we need to cover this technique they used called hematoxylin and eosin staining, or H&E staining. So what H&E staining is going to do is it's going to stain the nuclear components and the cytosol of the cell, a purple and pink color, which is a little bit similar. And it's going to leave everything that's hydrophobic, like our fats, our lipids, and our adipocytes, it's going to leave those clear. So going forward into the next few figures, when we see this H&E staining, for the most part, what's of interest to us is the adipocytes, which are going to be left clear. So we're not looking at what's stained, we're looking at what's not stained. Cool, so starting into figure 2, they're looking at brown adipose tissue and white adipose tissue, and they're looking at wild type compared to the SLN knockout. And what they're doing is they're dropping these mice down into cold crisis, which is 4 degrees Celsius. And what we can see it really well here in this white adipose tissue, and it's a little bit harder to see here in this brown adipose tissue, but here's a better figure. We can see that there's much less clear, there's much more pink in these bottom photos than there is in these top photos. And more pink and less clear area means less adipocytes. So our fat is being depleted, our fat stores are being depleted in these mice. And we can see that our white adipose tissue fat depots are especially being depleted. And the reason that the white adipose tissue is being depleted a little bit more heavily than the brown adipose tissue, especially in these SLN knockouts, is because we are browning this white adipose tissue. So we are depleting this, depleting the white, turning it into brown. And the reason we want more brown is because brown adipose tissue is what's responsible for thermogenesis through UCP. So if this mice with the SLN knockout is more heavily relying on UCP, it makes sense that we're going to turn our white adipose tissue into brown adipose tissue and then use UCP in order to generate heat. So starting on these bottom two figures here, these Western blots, this one on the left is a little bit hard to tell with the brown adipose tissue, but it's a really clear result here on the white adipose tissue that UCP1 is heavily upregulated in the SLN knockout mice, which if we think about makes sense. If we're knocking out the SLN path of thermogenesis, then we need to more heavily rely on the UCP path of thermogenesis, which is going to cause us to upregulate that protein. So figure 3, starting up here with 3A and 3B. So we can kind of see a common trend in both of these graphs that as our temperature decreases, our oxygen consumption is going to increase, which is to be expected. We're going to have to use more energy in order to keep ourselves warm, so we're going to need more oxygen, and it would also make sense that we're going to need more food, which is a figure a little bit further down here. But the only difference between 3A and 3B is that in the 3B they are actually controlling and normalizing these results by body weight. And we can discuss why they decided to do that once we get through the rest of these figures. So looking at figure 3C and 3D, all they've done here is they've just zoomed in on this 4 degrees Celsius result from the two graphs above. So looking at that result, this graph on the left, 3C, is actually not super helpful because it's not controlled by body weight, but what we see is that the double knockout mice actually seem to have less oxygen consumption when compared to the other 3 mice. But once we control for body weight, we see that the double knockout mice actually have statistically significantly greater amount of oxygen consumption than the other 3 mice, which is kind of an interesting result. And for the results to change like that is a good justification for them to normalize by body weight, and for that to be something they consider. Starting on figures 3E and 3.4, I kind of hinted at this a little bit earlier, but when we look at food intake among these 4 different mice strains, we see that the double knockout mice are consuming the most food, which makes sense if they're working the hardest to maintain thermogenesis. They've had UCP and SLN knocked out, that they're going to have to consume the most food. And when we look at the oxygen consumption at 4 degrees Celsius, correcting for body weight, we also see kind of similar result where the double knockout mice are consuming the most oxygen in order to maintain that thermogenesis. So moving on to figure 3F, we're looking at change in body weight here, and we can see that one of these strains of mice has a significantly greater loss in body weight than the other 3. And this may be what hinted to the researchers that they need to control this data above for body weight, is that one of these was losing significantly more, but the double knockout mice are actually losing much more body weight than the other 3 mice strains. And the reason for that, kind of similar to what we saw in figure 2, and I can go back up to that real quick, is that the double knockout mice are actually browning a lot more of their white adipose tissue in order to try and do thermogenesis, because they're in more of a cold crisis. They're having a more difficult time maintaining thermogenesis, and therefore it makes sense that they're going to use more of their fat depots, or they're going to use more of their white adipose tissue and brown more of that. So starting with figure 4, we're kind of looking at visceral fat pad weight, which kind of confirms what we were looking at in figure 3F. But these DKO double knockout mice are losing a lot of weight in their visceral fat pads, and we kind of see the same result with the UCP knockout mice as well. So a lot of that fat, a lot of that weight that's disappearing is actually, like we said, the browning of white adipose tissue. And then we can see here, this is kind of an interesting result, that the SLN knockout mice seem to be losing a lot of weight in their fat pads as well, but this was not statistically different, while the UCP and the DKO results were statistically different. And looking at figure 4B, we can see that there's a lot more pink in these bottom photos compared to the top photos. And remember that a lot more pink means less fat. So kind of consistent with what we've seen before, we're losing our fat depots in all of these mice. So looking at table 1, they actually found some interesting results when they were raising these mice at 22 degrees versus 28 degrees. So the mice that they were raising at 22 degrees, and when they were doing these crosses to get the double knockout and the SLN knockout and the UCP knockout, they observed that they were getting much less of the DKO mice than what was expected, almost like it was following non-Mendelian inheritance. But what was kind of strange was that at 28 degrees Celsius, the mice that were raised at a little bit warmer temperature, they seemed to get all of the expected values of Mendelian inheritance. So kind of thinking about what could cause this, the only difference between these two samples is the temperature they were raised at. And what actually is responsible for this is that at 22 degrees Celsius, it was so cold that these DKO mice, who were not very well prepared to deal with the cold or to maintain thermogenesis, actually a lot of times that DKO mutation that they put into the mice ended up being fatal. So it's not that either of these samples were not following Mendelian inheritance, or that either the genes aren't following Mendelian inheritance. It was actually that they created a lethal type presentation of this DKO mice or DKO mouse line. So covering figure 5 here, we can start with figure 5a and 5b. So what they're doing with this acute cold challenge is they're placing the mice into an environment that is 4 degrees Celsius in order to see how many remain after 8 hours. But looking at figures 5a and 5b, the only difference between 5a and 5b in this 22 versus this 28 is actually the temperature that they were raised or reared. This data actually shows us is that the mice that were raised at 22 degrees Celsius in general seem to have a higher body temperature when compared to the mice raised at 28 degrees Celsius, which is an interesting result. And the way that we can kind of logic through that or explain that is that the mice that are raised at 28 degrees Celsius have never really had to rely on those thermogenesis pathways, the UCP, the SLN, and so those pathways aren't very heavily upregulated. While a mouse that was raised at 22 degrees Celsius might have had to rely on that thermogenesis pathway a little bit more heavily and could more easily upregulate those pathways when put into a cold crisis. So the 22 degrees Celsius mice in general had higher body temperatures throughout this cold crisis and seemed to be better prepared to handle the cold challenge. On to figures 5c and 5d. So what we see here kind of backs up what we saw in figures 5a and 5b, and something that is a very small detail that might be hard to pick up if you're not aware of it is that figure 5c here is after 8 hours, and figure 5d on the right here is after 2 hours. And then of course on the left is 22 degrees Celsius and on the right is 28 degrees Celsius, the environment they were raised in. So what we see is that after 8 hours there were many more 22 degrees Celsius raised mice left in this cold crisis environment when compared to the 28 degrees Celsius raised mice. And the way they decided whether or not a mouse was remaining after this amount of time was that they removed any mouse whose body temperature hit 30 degrees Celsius.
So before we go into the next figure, I just wanted to go over some background information that we're going to need to take a look at this experiment really effectively. So right in front of us we have two different molecules. On the left is norepinephrine and on the right we have epinephrine. They have similar structures and they have similar names, but they do have different physiological origins. While norepinephrine is a neurotransmitter in the sympathetic nervous system, epinephrine is a hormone excreted by the adrenal medulla. While they do have slightly different functions and they have different origins, in terms of non-shivering thermogenesis, we can consider them two molecules of the same classification. They are both tyrosine derivatives called catecholamines, and they interact with beta-adrenergic receptors, which are a type of G-protein coupled receptor. When they interact with these receptors, they promote catabolism of glycogen and triglycerides, which makes them an important molecule in terms of non-shivering thermogenesis and cold acclimation in general. These catecholamines have an important role in non-shivering thermogenesis. They're released during cold stress, and when they do, as described before, they interact with these beta-adrenergic receptors, which are a type of G-protein coupled receptor, to stimulate the catabolism of triglycerides and glycogen. Described here in this figure is the breakdown of triglycerides as a result of norepinephrine interacting with this beta-adrenergic receptor. We've went over this in class, so it should look pretty familiar to you. But what's really important and different about this figure is that some of these free fatty acids are used to directly activate UCP1, or thermogenin as it's labeled in this case. Additionally, some of these fatty acids are used to provide electron carriers to power proton pumping, as UCP1 needs some protons in the intermembrane space to be able to generate its heat. Aside from these fancy pathways, catecholamines actually stimulate piloerection, which is the involuntary raising of hairs, and peripheral vasoconstriction, which is the narrowing of blood vessels near the skin surface. Both of these are strategies to prevent heat transfer from the core of the body to the external environment by just providing a little extra layer of insulation. Now, having all of this knowledge, the authors of this paper wanted to answer the question, why could double knockout mice survive cold adaptation rather than acute cold exposure? So they made an experiment. What they did was they collected urine at different stages of the cold adaptation, and they tested for norepinephrine and epinephrine levels. Remember, both of these are types of catecholamines. So what they discovered looking at these catecholamine levels was that the double knockout mice and UCP1 knockout mice both had incredibly increased catecholamine levels, especially at lower temperatures compared to the other two groups, that is the SLN knockout and the wild type mice. Now there's one common denominator between these two groups, in that both of these groups have no UCP1 gene. That means that these increased catecholamine levels are likely linked to the UCP1 gene. Now remember, the interaction of catecholamines with the beta-adrenergic receptors stimulates the release of free fatty acids, which can activate UCP1. Now this is very important, as without a UCP1 gene, we can obviously have no activation of UCP1. Likely what's happening is that in the brown adipose tissue of these mice, the cells are discovering that there's insufficient activation of UCP1, which is then causing the upregulation of norepinephrine and epinephrine to try to make up for this difference. Of course, it's all futile because there's no UCP1 gene at all. However, this still begs the question as to how these mice survived cold adaptation at all. This is likely due to the regulation of heat loss via piloerection and peripheral vasoconstriction, as well as increased metabolic heat. There are methods to generate heat other than UCP1 and SLN. Though this is true, these methods are not preferred, as they are extremely energetically expensive, as well as very inefficient. So why did we even read this paper? Why did the scientists write this article? So before this article, and even currently, brown adipose tissue independent thermogenesis is very poorly understood. However, throughout the course of these experiments, the scientists were able to identify a protein potentially important for muscle-based non-shivering thermogenesis. This protein is, of course, sarcolipin, or SLN. Now also, this study indicates a sort of functional adaptability between sarcolipin and UCP1, where one gene can be upregulated in the absence or inefficiency of the other. However, while one can make up for the other's shortcomings, both brown adipose tissue and muscle-based mechanisms of thermogenesis are important in mice. We saw in this experiment with the SLN knockout mice that defective thermogenesis can cause the browning of fat, and we also saw that it increases the energy expenditure when one or the other gene is knocked out. What the authors also observed in these experiments is that the loss of both UCP1 and sarcolipin is extremely energetically costly. Now, this has the potential to be used to combat obesity if we can do more studies and we can learn to harness this power in a healthy manner. So let's just take a little bit of an overview at this paper. Take a look at the big picture. So as a whole, we can take away 4 facts. First is that all endotherms or mammals use some combination of UCP1 and sarcolipin for non-shivering thermogenesis. Some animals may have more brown adipose tissue, and as a result, they would use more UCP1 for their non-shivering thermogenesis. On the contrary, some animals may have more red muscle and use sarcolipin for their non-shivering thermogenesis primarily. Second is the fact that the loss of either gene, UCP1 or SLN, is energetically costly, but it's not detrimental. The scientists found in this paper that the loss of UCP1 and the loss of sarcolipin was about evenly energetically costly, which indicates that in mice, they rely on each gene about the same level. Third, we learned that the loss of both UCP1 and SLN is unsustainable. Remember that the scientists had to pull out a lot of the double knockout mice from the acute cold exposure experiments early because their core temperatures were getting too low. Remember also that the metabolic requirements of these double knockout mice in the exposure to the cold was extremely high. They were losing a lot of white adipose tissue, which means that over time, this would be extremely unsustainable as it's just too expensive to keep up with. Finally, and perhaps most importantly to us humans, this study provides insight into non-shivering thermogenesis in mammals with little to no brown adipose tissue. That class, of course, includes us, but it also includes other organisms such as birds. Now, just to wrap it all up, let's look at the whole storyline of this paper. So the authors went into writing this paper and doing these experiments knowing that the mechanisms underlying UCP1 independent thermogenesis are not well understood. So they designed some experiments targeting the effects of knocking out UCP1 and sarcolipin in mice. And what they found was that the loss of both sarcolipin and UCP1 results in compromised thermogenic ability and severe sensitivity to acute cold exposure. Now remember that these double knockout mice were able to survive cold acclimation, where the temperature was gradually dropped down, but it was still ultimately ruled that this was unsustainable as their metabolic requirement was too high and they were losing a lot of white adipose tissue. Remember also that the loss of either sarcolipin or UCP1 was ultimately sustainable over time, though it resulted in an increased metabolic requirement for these mice. Using these results, the authors were able to conclude that sarcolipin-mediated thermogenesis is required for optimal thermogenesis, at least in mice, and is upregulated in the absence of UCP1. This also happens the other way around, where in the absence of sarcolipin, UCP1 is upregulated to make up for that difference. Now, the whole point of this paper was to find out what UCP1 independent thermogenesis might look like. So the whole significance of this paper is that sarcolipin is a crucial contributor to thermogenesis and energy expenditure, and it's definitely a candidate to consider for future experiments on non-shivering thermogenesis. These are all of the references that we used in our presentation if you guys would like to go check them out yourselves. We thank you guys for your time and your attention in this presentation, and we hope you enjoyed.